Today I'm going to be talking about hereditary cancer syndromes in colorectal cancer. Here are my disclosures. This slide summarises my grant and research support, Speakers Bureau honoraria and consulting fees. I am also an employee of Cancer Care Manitoba. None of these disclosures have an impact or present any potential bias for the purposes of this presentation. The learning objectives of this presentation are to describe the genetic basis of hereditary cancer syndromes, describe how hereditary cancer syndromes led to the concept of tumour suppressor genes, distinguish between germline and somatic mutations in cancer development, and delineate the contribution of hereditary and genetics to colorectal cancer in Manitoba. So a few definitions and basic concepts to start. So what do we mean by mutation? Well, there are two main types of mutation that we'll be talking about in this presentation. The first is known as a single nucleotide variant, or SNV, where the letters in the DNA code are changed with the substitution of one letter in the alphabet. Shown in this example here, a series of CATs, which encode for one amino acid, is changed to a CCT, and this changes the amino acid to proline from its original amino acid code. These changes distort the function of the protein or block a particular amino acid from performing a critical function such as phosphorylation. And many of these single nucleotide variant events lead to a loss of function of the protein. There are other mutations that change the function to a gain of function or change the function completely, but we won't be talking about that further in this presentation. The other ways that cancers can inactivate genes is through deletion. So rather than changing the alphabet code within the DNA itself, here we see a chromosome, and a piece of the chromosome that contains a particular gene of interest is deleted or removed during mitosis. And this also leads to a loss of function in that the one copy of the gene is completely removed and is therefore not present in the daughter cells when the daughter cells undergo DNA replication. Few definitions. So when we talk about hereditary, we are talking about a disease that's determined by genetic factors and therefore able to be passed on from parents to their offspring. A germline mutation is a mutation that occurs in the genome of an individual from the point of conception. So this can be inherited from the parents, or it can occur de novo during early development. But that mutation is then carried into every single cell of the individual at birth. A somatic mutation is a mutation that arises in a cancer cell that is not present in the germline. So remember that most cancers, in fact, if not all cancers, acquire mutations in their DNA as they evolve from an early pre-neoplastic lesion to a benign tumour cell to a malignant tumour cell. And that's what's referred to as somatic mutagenesis in cancer. And finally, tumour suppressor. What do we mean by a tumour suppressor? A tumour suppressor is a gene that, when affected by a loss of function mutation in the germline, increases the risk of cancer. And it's the study of hereditary cancer syndromes that are affected by these loss of function mutations in tumour suppressors that led to the concept of tumour suppressors in the first place. And that is why hereditary cancer syndromes are so important for understanding how cancer evolves and progresses. And the original tumour suppressor that was studied many, many years ago and really defined the field is the gene that causes retinoblastoma. Retinoblastoma is caused by a mutation in the RB1 gene, and the study of this disorder led to what's known as the Knudsen two-hit hypothesis, one of the most important concepts we have in germline and hereditary cancer syndromes. So in the cases of bilateral retinoblastoma, children are born with a mutation in one copy of the RB1 gene on one of the chromosomes. The other chromosome has a wild-type copy, so RB1 is functioning at 50%, which is sufficient for normal development. During replication of the retinal progenitors, a chromosomal deletion event occurs. This is the so-called set and hit, purely a somatic one in the retina itself. So then those cells are left with a mutant copy of RB1 and the other wild-type copy is deleted, accounting for the earlier onset and bilateral nature of hereditary retinoblastoma. In sporadic cases where the tumour is unilateral, in the germline, the children are born with a normal RB1 gene, 2 normal copies. The first somatic event is a mutation in the RB1 gene which arises spontaneously through random chance in the developing retina. And then subsequently, a second somatic event leads to deletion of the wild-type copy of RB1 on the other chromosome, such that the cells now have inherited, all the daughter cells will have now inherited mutant RB1, and the tumour then develops. So you can see here the difference in time of onset between a germline tumour suppressor event and a somatic tumour suppressor event. And this concept of losing a wild-type copy of a gene on one chromosome is also known as loss of heterozygosity. So you can see here in this depiction, there is a mutant copy of RB1 on one chromosome. In the normal situation, there'll be a wild-type copy on the other chromosome so that the cells would be heterozygous for that mutation. Deleting the wild-type copy leads to a situation where the cell only has one mutant copy, and this is known as loss of heterozygosity. This is also a very important concept in understanding how tumour suppressors work. So one of the most important familial syndromes that we know of in colorectal cancer, which we can now understand based upon the work of Knudsen and retinoblastoma, is the APC gene, which is responsible for familial adenomatous polyposis. The APC gene regulates a signaling pathway called WNT, W-N-T, which is an ancient developmental pathway first identified in flies from a gene called wingless, because the mutated flies had no wings. Losing 1 or 2 copies of this APC gene induces an abnormal growth pattern within the crypts of the colon. The crypts is where the stem cells of the colonic epithelial reside. And the wingless signaling pathway is typically restricted to the base of the crypt, shown here on the left. Losing the APC gene deregulates this wingless signaling, and the number of progenitor-like cells that are potentially able to give rise to a tumour expands within the crypt, shown on the right, with the increasing numbers of yellow cells. So the number of cells that are vulnerable for transformation into cancer becomes expanded as a result of inactivation of this tumour suppressor gene. So in familial adenomatous polyposis, patients are born with 1 mutant copy of the APC gene. Once again, not having the APC gene at all is embryonic lethal, but having 1 functioning copy is sufficient for most babies to be born normally without any obvious defects. So in this situation, in the germline, these patients have a heterozygous mutation for the APC gene. If you look at their colons, they're completely normal. However, with loss of heterozygosity or deletion of the wild-type copy of APC gene, which occurs randomly with age, these patients develop multiple benign polyps of the colon, shown here in this colonoscopy image. With additional mutations in other tumour suppressor genes, such as P53, there's a high probability that some of these polyps will advance and become invasive malignant colorectal cancer. So you can see here the pattern from normal colon to colon with polyps, which are benign but malignant precursor lesions, all the way to invasive cancer. But because the entire colon, and in fact the entire body, carries 1 mutant copy of APC, this happens very, very quickly. So the polyps typically begin by age 10, and often by age 40 or 30 to 40, these patients have developed significant numbers of malignant colorectal cancers. And this concept is known as multi-step carcinogenesis, which was really described by Bert Vogelstein at Johns Hopkins University, who really identified the progression model of colorectal cancer. So APC mutation, you have a normal colon. If you lose the other copy of APC through a deletion in the chromosome, you begin to develop benign polyps. But you need additional mutations in other tumour suppressors or oncogenes such as RAS for a full-blown colorectal cancer to appear. But because, as I mentioned, the original founding mutation is present in the germline, this happens in a very compressed time frame to young patients. However, one of the most important concepts from this hereditary model is that we know from genomics in cancer that the APC gene, which is responsible for hereditary colorectal cancer, is actually mutated somatically in 61% of sporadic colorectal cancers. And this is a very important crossover point between hereditary colorectal cancer syndromes and somatic cancer mutations, in that the same gene is mutated in older patients with colorectal cancer who do not carry the mutation in the germline. So in this model, the patients are born with 2 normal copies of APC. They have a normal colon. Through random chance, mainly through aging, because these cells are replicating constantly, an APC mutation occurs in some of the cells of the colon. But that's not enough to cause cancer. The colon still looks normal. Once again, if the other copy of APC is lost through deletion or loss of heterozygosity, polyps will then appear. But in this case, there are 1 or 2 or 3 polyps per colon, not hundreds, as you see in the familial adenomatous polyposis coli patients. And then with the addition of additional mutations in the cancer cells, colorectal cancer appears. But the time frame here is much, much longer. So most of us will acquire some mutations in our colorectal cancer, in our colonic epithelial cells with age, but it takes multiple steps, including deletion of the other copy of the APC gene, and then further mutations to cause the colorectal cancer. And this starts at the age of 50, and by 75, the incidence of colorectal cancer in the population goes up. So it's the same multi-step carcinogenesis model, but it happens more slowly because this is not a germline cancer syndrome. The other syndrome that is very informative with respect to tumor suppressors in colorectal cancer is the Lynch syndrome, or hereditary non-polyposis colorectal cancer, or HNPCC. This disorder is caused by germline mutations in 4 different DNA mismatch repair genes: MLH1, MSH2, MSH6, and PMS2. The function of the proteins encoded by these genes is to detect and repair mismatches which occur commonly and randomly during DNA replication. Because the colonic crypt is a high turnover epithelial system, the replicating cells frequently acquire these mismatches and they need to be repaired. If there is a defect in this system, the mismatches are not repaired, and this leads to an expansion of repeat sequences shown on the bottom right here. And that's what's known as microsatellite instability. So repeat sequences of the DNA are prone to be expanded and over-replicated in the situation where mismatch repair genes are not functioning normally. So, importantly, what's important about Lynch syndrome and how different it is to FAP is that there is no multiple polyp precursor lesions to be found. These cancers arise through a very different mechanism. Typically, the precursor lesions are adenomas rather than polyps. They're very flat and often very hard to detect through colonoscopy. The acquisition of microsatellite instability when one loses the other copy of the mismatch repair gene leads to acceleration of the tumor formation. But once again, additional mutations in genes such as TGF-beta, BAX, BRAF are required for an invasive full-blown adenocarcinoma to develop. So this is a very, very different model to what we see in familial adenomatous polyposis, and it is due to random accumulation of mutations based upon a mismatch DNA repair deficiency that is present within these tumor cells. So hereditary colorectal cancer has been very informative because it's helped us develop our understanding of tumor suppressors, but clinically it's also important because about 6 to 10% of all colorectal cancers that we see in the clinic probably have a hereditary association. In HMPCC, the non-polyposis hereditary colorectal cancer, which is most elegantly described in patients with Lynch syndrome, there are mutations in these mismatch repair gene families that I've described to you before. There are other hereditary polyposis colorectal cancer syndromes, including familial adenomatous polyposis, or FAP, which can occur in mutations in APC or MUTYH. And there are other less common syndromes, such as juvenile polyposis, Pearce-Jaeger syndrome, and Cowden syndrome, which are associated with germline mutations in more distinct, functionally distinct tumor suppressor genes. So the clinical impact, and one of the most important areas in which hereditary colon cancer affects clinical practice, is in taking the family history. Remembering that many of patients who have potentially hereditary colorectal cancer may not necessarily fit into the categories shown here. The other important thing to remember is that in both Lynch syndrome and familial adenomatous polyposis, there are other cancers that can arise in these patients for which they are at risk. Particularly in Lynch syndrome, endometrial cancer, gastric cancer, and pancreas cancer risks are also elevated. And in FAP, desmoid tumors, osteomas, epidermal cysts, pancreatic cancer, and other gastrointestinal cancers are more likely. The non-syndromic familial colorectal cancers are harder to understand and harder to map genetically, but you don't have to have Lynch syndrome or familial adenomatous polyposis to have a germline predisposition to colorectal cancer, because many of these mutations or alleles we still don't know about. So family history is very important, not just for colorectal cancer, but for other cancers as well. So the take-home messages are that hereditary colorectal cancer genes are somatically mutated in sporadic cases of colorectal cancer. And this is important because it's helped us our understanding of what a tumor suppressor is. A thorough family history is essential to identify potential cases, and hereditary contributions to colorectal cancer are probably more common than the known syndromes that we recognize and that I've described to you here today. Cancers other than colorectal cancer can occur in these germline syndromes, and once again, a thorough family history is needed to identify cancers that these patients might be at risk for outside of colorectal cancer. Thank you, and I'll be happy to take any questions.
So I guess, Neil, I guess I can ask a question. We have been able to make use of the mutations that trigger the hereditary non-polyposis colorectal cancer syndrome cancers. We can come up with therapeutic pathways. Is there anything being done with relation to the FAP-associated cancers? Does that lead to any sort of a therapeutic opportunity?
That's a great question, Piotr. Thank you. This activation of wingless signaling through loss of APC is a notoriously difficult pathway to drug. So people have been working on small molecules for many years, and it's really, unfortunately, been a very frustrating and fruitless exercise. But there is evidence, and this is a little out of my area clinically, but experimentally, there's very good evidence that aspirin or COX-2 inhibitors can reduce or actually significantly delay the formation of polyps and the conversion of polyps to adenocarcinoma in FAP patients. There's also epidemiological evidence that in the general population, low-dose aspirin in particular is associated with a reduction in colorectal cancer incidence, although there's not a direct causal link that's fully been established. So cyclooxygenase inhibitors do seem to have an effect on patients with the syndrome, but unfortunately in established colorectal cancer, it's been a very frustrating 20 years of experiments trying to develop small molecules that intervene in the presence of an APC mutation. And today, there's really not been very effective therapies for that.
All right. So if no other questions, I guess we can go back to the main link.
Great.
Thanks, Neil.
Thanks, everyone. Thanks, Piotr.